Thank you very much for inviting me to talk. And I know that Natalie and Michael will also be speaking a little bit about tools for unearthing roots from field to data. So I'll be focusing on comparing the root image analysis softwares that are available. So my name is Katie. I'm based at University College Dublin, and I'm supervised by Olaf Schmidt and Saoirse Tracy there, as well as Fiona Brennan at Teagasc. So why I look at roots, so I just introducing the perspective I'm coming from. My PhD project is focusing on managing plant diversity and plant soil biota interactions for nitrogen efficiency in Irish grasslands. So multi-species grasslands are a type of agricultural grasslands which have additional plant species and have been found to be high yielding under lower nitrogen fertilizer compared to just grass-only monocultures, which are quite common here. So they also, by introducing new species, you get extra benefits such as new functional groups. You might have nitrogen fixing legumes. You have different spatial distribution of roots in the ground, and they have different resource acquisition strategies. So you're getting that nutrient efficiency. So there's a big focus of roots in my research. So my process for analysing roots would be to take soil cores from the field. These are then washed, so you remove the soil, and so you're just left with the roots. And then these are scanned. So usually once you've got the clean roots, you can spread them out in a transparent tray in water, and then you can place them on a scanner. So this is very similar to the type of document scanner you might have in your office, and you're left with an image that you can see. And then these images can be analyzed using software. So these can detect roots, they can manage, they can measure different morphologies of the roots as well. So the challenge I had when I was analysing my root samples, I was sampling field plots that had monocultures and mixtures of 6 different species with very diverse root architectures in the same sample sometimes. So you could have very small, very fine roots, such as the grass species, which were less than 0.1 millimeters. And then you had species such as chicory, which could have roots that were bigger than 2 centimeters. So there's a big diversity. And why that was a challenge was that one of the most commonly used softwares that we would usually use in our lab was WinRhizo, and it's been found to have inaccurate volume estimates where there are heterogeneous root diameters. So because of this, I decided to compare some different root analysis softwares. So WinRhizo, as I mentioned, is one that some of you may have heard of before. It's one of the most commonly used software for root analysis and is used in a lot of publications, whereas Rhizovision Explorer is a relatively new software that was only released about 2 years ago. So WinRhizo is a commercial software, so it's also, because it's commercial and proprietary, it means that all of the mechanisms it uses to measure and analyze roots, we don't always know, whereas Rhizovision Explorer is actually open source. WinRhizo has a license fee, which can be quite expensive, whereas Rhizovision Explorer is open access and free to use, which makes it quite an interesting one to look at. And as I previously mentioned, there's different ways to calculate the root volume. So WinRhizo uses a method where it calculates based on an average diameter, which means that if you've got big differences in the types of diameters you have in your samples, it can underestimate the volume, which has been found in a number of studies. Rhizovision Explorer uses a different method where it kind of counts per pixel along the length. It will measure those diameters separately and adds it up, so it's actually more accurate when it's accounting for volume, and it means it's better for those samples you might have with more heterogeneous root diameters. So when I came to comparing those softwares, I looked at they've already been compared just generally, whether they're similar, but I wanted to specifically look at, are these estimates affected by the plant species or the presence of multiple species with different root morphologies in the same samples. So I took soil cores from field plots. So I had monocultures of 6 different species. So there was 2 grass species, 2 legumes, and 2 herbs. I had a 2 species mixtures of perennial ryegrass and white clover, as well as a mixture of all 6 species, and these were separated into 3 soil depths. So the same scanned images that I got from the scanner were analysed by both softwares using the same settings in order to compare them. So when I was comparing them, I could see that both were highly correlated. In my graphs, I have Rhizovision Explorer on the y-axis and WinRhizo on the x-axis, and the dashed line shows where the 1-to-1 ratio would be. So points that are on that line would be the same estimates for both software. So you can see that there is variation. Generally, WinRhizo, the volume and length estimates were significantly lower in the top 10 centimeters of the soil. So this would be where we'd expect to have the biggest difference in root diameters, as well as just a lot more roots from that section of the soil. You can see in particular in terms of root volume, there's certain plant communities which are different. So we have the chicory and rye clover, which are much lower estimates in WinRhizo, which is what we would expect, given that those were the species that had the biggest kind of thickest taproots, and so they had the biggest heterogeneous in the samples. We could see the root diameter samples where the estimates were different in all depths. There's a lot of variation in root diameter. Whereas root surface area overall, there's no significant difference. So I wanted to understand where these differences lie within the types of roots. So both software have a feature where we're allowed to split up the roots into diameter classes. So here I split them up into 10 diameter classes, with the smallest diameter roots in the dark purple color, and as it gets larger diameter, those are the lighter colors to yellow. So we can see that the total root length, so the total root length here assigned to each diameter class was compared. The largest differences were in that smallest diameter classes. So this is particularly due to WinRhizo attributing more roots to that small diameter class. It's particularly evident for the grass species, so perennial ryegrass and timothy grass, which are in the first 2 panels, had very large differences. So that's contributing to a big difference in these 2 softwares. So how do we know which software is correct? Both of them just provide estimates, they're not true measurements. So we need a way of comparing them using something with known dimensions. So we decided to use fishing line, because you can get clear fishing line, which is quite similar to colorless roots. You can get it in a number of diameters, as well as being able to then color it if you want to mimic stained roots. So we used a clear fishing line that was 0.14 millimeter diameter, so that would have fitted into our smallest diameter class. And we could compare to the known measured lengths, as well as the known measured diameters, to what the software were estimating. So here, the dashed line would show where the measured length and the estimates would meet. So points that are closer to that are more accurate, where if they're further away, they're less accurate. So you can see that the Rhizovision Explorer in pink is generally higher than the true length, whereas WinRhizo is lower than the true length. There's higher error rates for Rhizovision Explorer, and there is more accurate estimates for fishing line that was colored black. So those are the square points. You can see that those are closest to the line for both software. So that could indicate that, especially for those very thin roots, staining would make accuracy in terms of length estimates better. So why were there length estimates? So we saw that at the thresholding setting that we chose, Rhizovision Explorer was processing some of those clear fishing line as two separate roots. So we're using the 220 thresholding setting, but at 230, this was more accurate. So altering the thresholding is an option. This was pointed out by Larry York, who's one of the creators of Rhizovision Explorer. We could also see that when rhizo in some of our root samples was not recognizing some of the fine roots that rhizovision did. So you can see in some of the middle sections, there's parts that when rhizo is skipping. So this could potentially explain why there is a low underestimation of root length. So we also looked at the diameter estimate. So we used a microscope to measure the true diameter of the fishing line. So the dashed line shows where that is. And we could see the rhizovision in pink was a lot closer to the true diameter than with rhizo. The accuracy was affected by when we cut the fishing line into smaller fragments, as well as when we colored it black. And in the case of when rhizo, it was almost doubling the estimated diameter when we colored it. So that might be an issue if you are staining. So what can we take from these differences? Both software give generally similar root rate estimates, especially where roots are more homogeneous. So we didn't see large differences in the lower depths. So if you have species which are have very similar root traits, they kind of root morphologies, you might not see a big difference. But we saw that even under the same settings, both software can act differently. So in the case of Wind Rhizo, it was underestimating root length and volume, and Rhizovision Explorer could overestimate some fine root lengths. So some tips for how to get more accurate estimates from these software, whichever one you're using. You can increase the image quality. This is especially important for root diameter. However, it will take longer to actually acquire the images and analyze them. So it depends how many samples you have and how feasible it is. You can check your thresholding settings. So make sure that you can check that the very fine roots are being recognized correctly. So we saw that with Wind Rhizo, sometimes they weren't being recognized at all, whereas with Rhizovision Explorer, they were being recognized as 2 roots or there was only 1. Staining your roots is another option, so it improves contrast, especially if you've got very fine roots such as grasses, but it may limit chemical analyses you can do, so you might have to have a subsample for this, and it might also affect the accuracy of diameter estimates, but this would hopefully be mitigated by using a higher image quality. Another method is just to minimize root crossover in the tray with scanning. So this is a good practice because obviously if there's a lot of overlap, it makes it harder for the software to recognize where the roots are. So most of this research was taken from my publication in the Plant Phenome Journal. So I've got the links here, and I've also got some other resources in terms of tips for using the software, doing root washing, and I'm happy to share all of these because I know there's a lot on the slide. So thank you for listening. Welcome any questions or comments. And I would also like to extend my thanks to my supervisors and the research groups at Teagasc and UCD. So I'm happy to do questions now or maybe after the others have talked, whichever you prefer.
Oh, super. Thank you, Katie, so much. It was very interesting. I don't know if Michael König and Nathalie Oram are also presenting, or yes, maybe we do the presentations first and then continue with questions. Otherwise, we will have 3 hours of virtual academy. Michael or Nathalie, who is continuing?
I think Michael, do you want to go ahead?
I tried to share the screen. Can you see it?
Yes, now.
Yes, walking. That is a real Wind Rhizo.
I would like to talk about some personal experiences with the second program, the Rhizovision Explorer, and I used within my thesis 8 plant species from 2 different communities, and they all have different root structures. And we took root systems from always 3 plant individuals per species. We weighted them fresh and stained them in ionized water with a neutral red. And then we washed them to remove the additional color. And then we used a normal typical scanner. It was the Epson Perfection V700, but of course any other scanner is also fine. Just it's important that there's enough space to put a plastic tray into the scanner so that there's enough space for the water and the roots, and of course for safety issues. And then we use the software that comes with this scanner, and we scanned the roots at 600 dpi. And this is the software from the scanner. This is nothing difficult, and yeah, with 300 the quality is, in my opinion, not good enough. But over 600, of course, the quality improves, but it takes a lot of more time. And I think 600 is in many cases enough, but this depends then also on the research questions. And I would recommend you also to literatures about the program for the Reservation Explorer. And this is how the scans look like. Here we used a different color. This is a simple Pelican black dye. So a cheaper version, and for some plant species it worked quite good, for others less good. And this is a negative example because if there are air bubbles within the scanner, or if there's some water on the glass above, then the scan will not be good enough, and the program will definitely not be able to detect here the roots and analyze it correctly. So I would first check a simple dye, and if it works well for all species or only one species, then I would go for it, and otherwise search for other colors. And for the settings in the program, we chose 600 dpi because you need to insert this. Then we started a broken roots analysis, and the root pruning threshold we set to 10. This was recommended by the literature, and we filtered the non-root objects with a size of 1 pixel. And I would definitely recommend to play a bit with these numbers, also to understand it better and how it affects the image. This is very interesting. And then the program calculated the total root surface area, the root length, root volume, and the average root diameter. And there's also an option to set additional settings so that it calculates this data regarding different thresholds, for example, diameters between 1 and 2 millimeters, 3 and 5, and whatever one likes to know. And here I did this with different settings, and each color is for a different diameter section. And on the left side, there is the scan of Plantago lanceolata, and on the right side, there is the analyzed picture. And the program, I think, is very good. It also detects where they were small and thin roots, also these pieces, and it removes the dirt if there are very small pieces left that were not washed away by the cleaning before. And then because we weighted the root pieces afterwards, we estimated the dry weight to identify the specific roof area, specific roof length, and the root tissue density. And I imported the data that the program provided directly and very fast, and downloaded it as an Excel sheet. I think this is very useful and easy also. And generally the program was, at least in my case, very fast. I think it ran 100 images within some minutes, definitely less than an hour. This depends, of course, on the computer, but I think I was very surprised about how fast and it delivered all the data. And thanks a lot for letting me talk to you. And Natalie would also have some feedbacks about our work.
Thank you, Michael.
So I'll just share my screen now. So can you see the full screen edition?
Yes.
Okay, cool. So so my name is Natalie. So first, thanks for having me. So I'm a postdoc, a Marie Curie Fellow, and I'm between the University of Innsbruck, where I worked with great students like Michael and Czegisk. So my whole project is looking at droughts and basically how drought legacies, or we call them soil memories, affects grassland resilience. And so I look at things like the interactions between plants and microbes and carbon transfer and nitrogen transfer between these two groups. But today I'll talk about something very technical, which is before all of this, which is how to make a root washing table. So it sounds kind of simple, but getting the roots out of the ground can be quite a hassle. And for instance, when I arrived at Innsbruck, we were doing it kind of over buckets. So this doesn't work so well because you have to be comfortable for many hours when you're washing the roots. So we built one in Innsbruck that was just kind of a MacGyvered one, but in Chagos we got a chance to build a really nice one. So and I did this with Louise, whose picture is here. So this is like our basic drawing of a root washing table. And so it has a grate, and then the water filters in to a bucket, and then this can level kind of drain out, so the soil's at the bottom and the water can run off. So this is our table, and basically if you are interested in building a root washing table, I've done this a couple of times now, and I'm very happy to share any experience I have or drawings. But maybe this could be more of a discussion thing. But this is our gorgeous table, and it makes it actually quite easy to wash roots and much more enjoyable than sitting over a bucket. So that's actually all for me. So if you have any questions or anything, I'm happy to take them.
Wow, super. Thank you, Natalie. Thank you all for those really nice presentations. I think we have a lot of questions, I'm sure. We already have one in the chat. Thank you, Michael. Nice talk. How do you manage to get roots of different diameter dyed with different colors?
Thanks a lot. You mean how we chose the dyes, or?
Maria Hernandez Soriano, can you specify this question?
Yes, thank you. I was wondering how you manage to get the different diameters color with different dyes. Kind of, how do you do it?
Yeah, actually, it didn't work so well for each species with the Pelican Black because it had some issues with the very thin roots for some species. But for others, it dyed all of it quite well. And yeah, it really wasn't that difficult. Maybe I can share you also some information about our protocol. With my supervisor is also okay with that. If you're interested into the
very much. I'm just starting to work a bit with Rhizovision, and it sounds very interesting in the different dyes. So if you could share a protocol, that would be great. Thank you.
Yes, Michael, if you could send it to me, I will share it and spread it. Would be nice. Thank you. Cool.
We have to make sure that we credit Michael, though.
Yes.
We always credit him.
Any other questions? Becky?
I have a question. I was more to Katie. Thanks for a great presentation, all of you, actually. It was really nice. It's great to see something so practical. Katie, which system did you prefer to use in terms of not being the best technical person in terms of computers? Which one did you say is the most sort of intuitive to use? Which system? That's what would more interest me than anything.
Yeah, no worries. I don't know if there's much difference in terms of intuitively. I think both systems, there's kind of, once you read the documentations, you should be able to follow it. But at least with our version of WinRhizo, it was very slow to analyze. So sometimes we would have to leave it overnight, whereas I think Michael also mentioned with Rhizovision Explorer is very, very quick. So that's one kind of benefit. I don't know if it's just because we have an older version, but yeah, it definitely has to, it would definitely took a long time. So that would definitely be a benefit. And I think there's a lot of the creators of Rhizovision Explorer have a lot of videos and things that you can follow, which also makes it a bit easier to learn, I think.
Great, thanks.
Any other questions? I think there was one in the chat also. RiserVision is a new program for us to use. Is there a guide tutorial? Ah, okay, I think you already answered that. And
I got one. I don't know if you have other.
Isabel?
But I got a question is coming back to the first presentation, Katie. When you're comparing WinRiso and RiserVision, maybe I missed the information, but what was the resolution you were working? Because in the talk just after we had 600 dpi, was it the same? Did you try several resolution and after did again your relationship or not?
Yeah, so I also use 600 dpi. I think a lot of people use that resolution. We did test with the fishing line at a number of different resolutions. There was, I think in both software, there were improvements, which was good, but I think beyond 1200, the computer starts to crash when it's analyzing it. So I think even from 800 to 1000, you see improvements, yeah. But I don't think there is, I can't remember if there's big differences between the software still. I can double check for you. I have the graph somewhere.
Thank you. So we have 2 more minutes. Do we have any other questions? I think there was also one question in the chat about the mesh size of the root washing device. Natalie, do you want to tell us something about it? Or
Yeah, so for your root washing table, we actually just bought a commercial grate. It's also the grates you see sometimes on sidewalks or like industrial stairs. So this is like 2 centimeters by 2 centimeters, but on top of this, you use your like root washing kit. So I have a sieve that's I use like 0.2 millimeters because I'm interested in the very fine roots. So then I wash like through the sieve and then it goes to the table. But the table is nice because it's just somewhere you can sit and then just wash into it, let's say. Yeah.
All experienced root washers will be very jealous of your
Yeah.
So we're always looking for volunteers for the Virtual Academy. I think it's a really good forum to sort of showcase your work. And so if anybody wants to, please contact Anna. Everybody's got Anna's.
yes, please do.
Thanks so much again for our 3 speakers. It was really excellent presentation. We've learned a lot.
Yes.
Thanks for having us.
Thanks so much. And yeah, see you at the next Virtual Academy in 1 month.
Thank you.
Thank
Have
you.
a nice weekend.
Bye.
Bye-bye.
Ciao.
Thank you very
Bye.
much.